length. For your lab reports, they should be nice and beefy introductions. But when you read this introduction, ask the following questions. What's the big questions of the field? And so this paper is about circadian rhythms, it's about sleep cycles, and so the field itself is trying to answer what are the external factors that affect sleep and wake cycles? What goes into the algorithm that tells you you need to sleep now or you need to wake up now? That's the big kind of overarching question of the field, not just the paper, but the field that this paper resides in. Then summarize the background information. This introduction gave you a whole bunch of background information. What is it telling you? And so in this instance, it's telling you that there's many biological processes that are activated and inhibited by a circadian rhythm. Circadian rhythm kind of tells your body, is it daytime, is it nighttime, and it adjusts processes accordingly. And light is a key external factor that affects your circadian rhythm. Everyone knows that when you travel the world, you change your light cycle, and so that throws your circadian rhythm off. And so that's kind of what they're talking about. And they're saying, well, a lot of people use phones, tablets, and computers, and those are also light-emitting objects, and those also are used at nighttime. So what's the effect of that? And that kind of gets you at the bigger question. What's the specific question this paper is going to answer? And it's does reading screens or print form affect the circadian cycle? So just reading a paper with light shining on it or reading from an iPad going to be significantly different from one another when it comes to reading at night, doing anything at night. And so what's their approach? Usually in the introduction they give you a brief analysis of what's going on, and so basically the idea is one group is going to read with ebooks, one group is going to read the paper in light. They're going to measure melatonin levels. Melatonin, if you didn't know what that was, go Google it. It's basically the protein that basically induces sleep. And they're going to do something called a polysomnographic recording, and if you didn't know what that is, again, Google it. It has its own Wikipedia page. It's basically brain recording, brain electric recordings while you're sleeping. It's called poly because it's more than one kind of reading. Somna relates to sleep, and graphy means collecting data, basically. And then they're gonna give objective and subjective tests of sleepiness. So basically, they're gonna do an objective test where they rate how groggy someone is after they wake up by having them do alertness test, and then they're gonna actually ask them how groggy do you feel? That's the subjective test. That's great. So you kind of already have an idea about what's going on and how they're going to do it. Then you should read the mitosis method section, and as you do it, you want to diagram out the methods and materials. And so I kind of just did that right here. I said 12 people are going to be split into two groups. One's going to be reading with paper, one was going to be reading with an iPad, and they're going to do that 4 hours before bed. And then while they do that, then these things are going to happen. The polysomnographic readings are going to happen the last 2 hours before and after sleep, and then the sleepiness test is going to happen. And then they're going to have also, along the side, they're going to have hourly blood samples so they can get at these melatonin levels. So this is just you internalizing what's happening. So graph it out, because some experiments can be really complicated. They have 3 different experiments, and they're collecting 6 different variables. It can get really complicated to sort it all out. So just diagram it out. It can be a flow chart, it can be a cartoon, it doesn't really matter, as long as you find a way to internalize it. And of course, as you get more familiar with techniques and physiology and something like that, just reading the materials and methods section is easy. You won't have to diagram it out, but in the beginning it's really important to do that.
Do you have to share an article with you first?
Nope. You do have to also submit the PDF of the article, just to show that you actually physically read it. Some people just read the abstract and try to do the whole summary just from the abstract.
It's an article that actually exists.
Yeah, you gotta show me that you actually were able to, you had the ability to read it, and hence you had the PDF for it. Anyway, so then go through the results and try to summarize each experiment they did, each thing they tried to collect, experiment the conclusion of the result they got. I said they measured melatonin levels, and oftentimes you can relate this experiment to a figure. So you're not only summarizing the experiment they did, but the figure they associated with it. And so they found that melatonin suppression was greater in iPad users by with a 1.5 hour delay. So melatonin was only really being seen in the bloodstream at significant levels an hour and a half after the people were just reading a piece of paper. You look at subjective surveys, people who were iPad users actually did feel took longer for them to fall asleep, and they said themselves that they were groggier in the morning. And the objective tests confirmed that. They said specifically that they fell asleep on average about 10 times later. And iPad users had, and this is the polysomnography, this is basically all the brain readings they're getting. They found that iPad users had less REM sleep, but most other parameters didn't change. This is step 4. Tons of mistakes in this one. Step 4, you read the discussion and you ask yourself, what are the interpretations of the results? So given all these results, what do they think about it? How does it answer the question? And the idea is you make your interpretation first, and then you see how the authors make their interpretation. And in the beginning, when you're starting out and reading papers, it's totally okay for you to first start out with a different opinion, but then when you read the author's opinion, you change your mind. That's totally okay. They're the expert. You're not. So if their interpretation changes your mind, that's totally okay. But you have to be aware that as you go on, get a little bit more familiar with your topics, with your fields, not always believe the author's interpretation. They might be biased. You may think this data is not as strong as they're claiming. Various things like that. So everything I talked about is highlighted in your supplemental PDF. It's there, page I don't know. It's one of the tabs. Just look it up. It's basically label how to read and understand a scientific article. I just basically talked all about all the steps that go into it. So go check it out. Spend some time this week. Go find a paper you're interested in, and start reading it. It's going to take some time to digest it and make it fun. Find something that's interesting. I always get a kick out of reading your guys' journal summaries because I get to learn a whole bunch of stuff, and it's always about really interesting topics. Like one was about thermoregulation of students who have to take tests, and it shows that students heat up like crazy when they take tests and stuff like that. Things like that. It doesn't have to be super serious science, but it has to be science. It has to be physiology related. All right. Oh yeah. That's a copy slide. All right. Any questions about that? No? All right. So let's get on with the topic at hand. So sorry about not posting the slides earlier. I really thought I posted them earlier. I checked at 10 a.m. today that they weren't posted. So if you ever see that, like the night before or something, just shoot me an email, and then that'll help me. But I usually try to get it the night before. So we're going to talk about cell signaling and signal transduction. So this is basically the idea of how cells talk to each other. And this is a huge thing in physiology because homeostasis is primarily one of the largest goals of the body, is to keep things in balance, to keep things running smoothly. And to do that, that takes coordination, right? That takes coordination of cells, that takes coordination of tissue, and to do that coordination, they need to communicate. And so cells need to find a way to talk to other cells to maintain this order. And so oftentimes it's one cell telling other cells to do stuff. And how they communicate is going to basically depend on the distance between them. If it's close, they're going to rely on methods of diffusion to talk to each other. But so yeah, if it's close, they're going to primarily rely on diffusion. But if it's far, diffusion's not going to work. Diffusion's way too slow. If I had to wait for a molecule to diffuse from my brain to my kidneys, that could take like a day, right? I could die by then, right? So you had to find some faster way. And so things that are far away, we would have to use the bloodstream. Now, keep in mind that there's still some diffusion at play here, but it's not the primary way of transport here. And so yeah, a signal has to go from one cell to another, and this signal, of course, is going to be some kind of ligand. The word ligand hopefully sounds familiar. Whenever we talk about proteins and their interactions, there's always the protein, and whatever it interacts with is called a ligand. And so, just like any other protein interaction, anything that involves signal molecule is the ligand, and whatever protein that's going to be the receptor to that signal is going to be the protein of interest here. And so when it comes to short distance signaling, it comes in 3 different flavors, and they all serve their various purposes. The first one is just to have gap junctions. Hopefully you remember this from anatomy, but cells can have gap junctions between themselves. That's basically what it means, is they have these bridges, these what we call cytoplasmic bridges that connect the cytoplasm from one cell to the other cell. So they basically allow cytoplasmic content to diffuse from one side to the